to frame that discussion about the impact, to frame that discussion about the use of genomics and really taking genomics to one or many steps forward. And to frame our discussion today, I'm very, very pleased to introduce Dr. Brendan Frey, our keynote speaker today, CEO of Deep Genomics. So Brendan Frey is an internationally acclaimed entrepreneur, engineer, and scientist. So imagine that combination. He is a co-founder, CEO, and chief engineer of Deep Genomics, and the co-founder of the Vectoral Institute for Artificial Intelligence. As a co-founder, Brendan led the launch of Deep Genomics in 2015. Imagine 2015, we're talking here 2015, talking about genomics, artificial intelligence, and he created and launched Deep Genomics to address the need for a new data and AI-driven approach for genomic medicine. Brendan has made fundamental contributions in the field of deep learning, genomic medicine, and information technology. He has co-authored over 200 papers in these areas, including over a dozen that appear in Nature, Science, Cell, and were on multiple occasions highlighted in the front cover of those magazines. His inventions include one of the first learning algorithms, factor graphs, a coding method that is used in most telecommunications devices, affinity propagation, deep learning models for splicing, protein, DNA and RNA interactions, and polyadenylation. Brendan holds numerous distinctions and is professor of engineering and medicine at the University of Toronto and a senior fellow of the Canadian Institute for Advanced Research. So I think I might not have covered everything, Brenda, but I think we're all super excited to have you here with us today, and we are very much looking forward to your presentation.
Thank you very much, Catalina. So I'm very pleased to be here today, and thank you to the organizers for inviting me. As Catalina mentioned, I've been part of the genomics community in Canada for many years, all the way back to 2002, 2003, when I first started becoming interested in the genome. And really, I guess my story, in a sense, is very appropriate for this session because I was a researcher in genome biology between 2002 and then 2015. And at that point in time, the machine learning and AI systems that we had built in order to unlock the secrets, really, of genome biology were working so well that we decided to spin out a company, Deep Genomics. And so, you know, I've been on both sides in terms of just pure research for a long period of time, and now fully focused on translation, essentially. So hopefully I have some perspectives that are helpful, and I'm pleased to share those with you. So I'm going to share my slides, and usual technical delay here while hopefully this works. Can you see those?
Yes.
Perfect.
Good. Now we see it, yeah, full screen. Thank you.
All right, wonderful. All right, so as Catalina mentioned, I'm the CEO and founder of Deep Genomics. And just to give you a bit of context on the company, and then I'll dive right into it, but a bit of context on the company, founded in 2015. We've raised roughly $300 million in funding. And all of our investors are behind the whole concept of Deep Genomics, which is decoding the genome to create life-changing therapies. And what's key here is that, you know, when you think of biotech, a lot of biotech companies are built to make one or two drugs, right? Or test out a chemistry and then maybe make one or two drugs. But I think everybody working in the genomics community is really doing it because they believe there's much more to it than that, one or two drugs, that really the genome is a resource of knowledge that will propel our understanding of our biology for centuries to come. And that's really the foundation of Deep Genomics. That's why we exist. You know, if we made one or two drugs, that would be great for the patients we serve, but it would not really be the goal of the company. The goal of the company is to really industrialize the productions of medicines to help patients in need. All right, so here's a quick snapshot of the team. We've got lots of different backgrounds, of course: machine learning, computational biology, software engineering, experimental biology, chemistry, drug development, business development. Actually maybe kind of reflective of the kinds of teams that you would see at a typical research group that's working on the genome. But we've recruited people, of course, from the from our surrounding. One moment, I need to clear my throat. There we go. So we recruited people from the surrounding environs, of course, like just amazing people in Canada, largely due to a lot of the funding that Genome Canada and other funding agencies have provided over the last 15 years. But also recruited people out of Moderna, out of Facebook and places like Stanford, Shire, drug development companies like Shire, rare disease drug development companies, as well as companies that are developing bigger drugs, bigger programs like Pfizer. So an amazing team, and everything that we've done would not have been possible, of course, without this team. So I just want to note that. Now, our headquarters are in Toronto, and I think this is important, right? A lot of biotechs, they might get going in Toronto or get going in Canada, and then they move to the US, or they get acquired by a big company like Pfizer or something like that. So Toronto is our home. Toronto is the headquarters of Deep Genomics. We do have some remote sites, like we have an office on Kendall Square, for example, but we are based out of Toronto. And so, in terms of translation, I think that's another thing to keep in mind is when Genome Canada and other agencies are thinking about data sets they could produce or key personnel they could produce, are they okay with that flowing into the US, or do they want to use it to generate innovation within Canada? And I think that's very important to keep in mind. So here's our team. I won't spend a lot of time on this, but amazing people that have, like Ferb Massari, our Chief Medical Officer, over 30 years of experience developing drugs at places like Shire and Merck and Pfizer. We've got great strategic advisors. Tal Zaks will be announcing next week that Tal Zaks has joined us as a strategic advisor, extraordinary scientific advisors and board members as well. So the starting point of Deep Genomics is really genome biology. And of course, that's where the genome leads to, right? We sequence genomes because of genome biology. And one thing we've seen in the last, well, I guess in the last couple of years, particularly of note, but really over the last 10 years or so, is that RNA therapies have come to be thought of as just really disruptive therapeutic modalities. What's cool about RNA therapies is that the therapy is a sequence of letters. Okay, so it is digital information, if you like, whether you're talking about an oligonucleotide or an mRNA therapy or a vaccine, or you're talking about gene editing with the guide RNA, the guide template, the therapy is a sequence of letters. Now, there's chemistry, of course, wrapped around that sequence of letters to get into a cell. But once it's in the cell, that sequence of letters can target any gene. That sequence of letters can be changed to increase gene expression, produce more protein. It can be changed to decrease gene expression, produce less protein, or even can be used to change which isoform the protein produces. So these RNA therapies are an incredible digital toolkit for manipulating biology. What could be better matched to the genome than that? So Deep Genomics, our approach is again very linked to Genome Canada and a lot of the projects that you all work on, which is that we have a digital RNA biology platform. So it's all about RNA molecules, DNA, of course, and proteins. And when you think about RNA biology, so maybe think about all the RNA-seq data sets that you could produce, as well as the DNA sequencing data sets. There's over 100 petabytes of that kind of data publicly available. 100 petabytes. I started working in the field, as I said, in 2002, and since then, the amount of data has grown 1 million-fold. So just an incredible growth in the amount of data. Now, I think we all kind of agree that all the opportunities for understanding genome biology and also developing or understanding and developing treatments for genetic disorders, whether rare or common, somehow all of that does lie within this genome biology data, right? The opportunities are there. But the thing is that humans cannot do this. Humans are not up to the task of directly analyzing this data to find the opportunities. What's needed is an automated system that can pore over that 100 petabytes of data and all the new data sets that we'll be generating in the years to come, and find the best targets, the best mechanisms, and the best molecules to produce therapeutic interventions. And that's what Deep Genomics does. We build AI systems that can do that. And that enables us to actually imagine programming RNA therapies for almost any gene in any genetic condition. So we manipulate that sequence of letters. It's like writing a program. I'll just briefly kind of set the groundwork for the framework that I'll talk about, very related, of course, to any strategy that the Genome Canada community might take for impacting biology and whether it's translation to industry or impacting biology at the level of research. It's a central dogma. So we go from a gene to pre-mRNA to RNA to mRNA to protein. And what's really cool about this framework here, which is why I put the slide up, is that compared to other areas of investigation, so for example, pathophysiology or electronic health records, or even systems biology, okay? Compared to those other frameworks, RNA biology is quite well understood. We know about the different processes and systems that are involved here. When we have a pre-mRNA that will get manipulated through splicing and polyadenylation and microRNA stabilization through various processes that we know quite a bit about to produce the mRNA molecule and the protein, okay? So this kind of biology is well understood enough that we can draw a block diagram or a wiring diagram. And the same cannot be said for pathophysiology or for electronic health records or for other kinds of data sets that we might gather. Not to say that those aren't valuable, it's just a lot harder to interpret them because there is not a well understood framework. In contrast, for genobiology or RNA biology, we have a very good framework. So we can imagine mutations that cause disease, they're digital. It's a mutation to a DNA sequence. We can imagine all of these operations that cut and paste sequences of RNA to produce the mRNA that makes protein, that's digital as well. We know what the wiring diagram looks like, and involves sequence manipulation. RNA therapies, which is what Deep Genomics develops, they are also a sequence of letters. So we've got digital targets, we've got digital biology, and we've got digital therapies. It's an extraordinary opportunity for this community. One comment on the types of therapies we develop. They're steric blocking oligonucleotides. An example would be Spinraza for spinal muscular atrophy, roughly 18 to 20 nucleotides long, some chemistry to make sure it gets into the cell. But essentially that 18 to 20mer binds the mRNA molecule or the RNA molecule and changes these different processes that I mentioned before. And so, for example, it could alter splicing so as to produce some sort of therapeutic benefit. Now, just a quick snapshot of one example of the capabilities we have at Deep Genomics. There are dozens and dozens of different possibilities. And every day we're doing different things with those predictive systems, those machine learning systems. But I just wanted to highlight one example. If you were to rewind the clock, say 15 years before Spinraza and the target of Spinraza was discovered, and you were to scan the entire genome, so this is what's relevant to the genomics community, scan the entire genome and ask the question, where and are there exons such that if those exons were strengthened with a therapeutic compound, we could remediate a Mendelian disorder, okay? So if you ask that question, molecular biologists could not look across the entire genome and try to answer that question. You obviously need computational techniques, you need data analysis, you need AI. So we did that with our current AI systems, scan the entire genome and ask for each exon, if we were to strengthen that exon, would we remediate a disease? We found over 1,000 different therapeutic targets, potential therapeutic targets. And in the top 10% of that list was SMN2 exon 7, which is the therapeutic target for Spinraza, for spinal muscular atrophy. And all of this was done in one afternoon on a computer. Okay, so that's the power of genomics complemented with AI and bioinformatics and computational biology. It enables you to do that kind of thing. The other programs, by the way, we moved into our wet lab at Deep Genomics. We have over 30 people in an experimental facility right now. The number is growing. 10 of the programs we moved in there, 7 of them we validated the target. And so also genomics plus our AI systems is giving us a very high success rate in finding novel targets and novel mechanisms. That's not something you'll see in traditional pharmaceutical companies. But we did even more than that. As the panel at the bottom shows, we then focused on that target, SMN2 exon 7, and then in the computer, we evaluated 2,000 potential steric blocking oligotherapies. For each one, they're shown on the x-axis in the plot on the lower right. For each one, what I'm plotting here is the predicted effect on strengthening the exon. So that would produce a therapeutic benefit for each of those steric blocking oligos, computationally predicted. And so the peaks would correspond to compounds that are predicted to produce therapeutic benefit. You can see that Spinraza is one of those peaks on the right there. So that's the sequence of letters that is Spinraza. And the other peaks correspond to other compounds. When we took those into our wet lab facility and checked them, we were able to achieve higher efficacy in vitro than Spinraza itself. So not only do the AI systems find Spinraza, they can also find compounds that work better. Just briefly, I won't go into detail on this one here, but we can also apply our systems to discover novel complex disease targets. So spinal muscular atrophy is rare disease, but we can also look at complex disease targets. So if we have available the appropriate whole genome sequencing or whole exome sequencing data sets, we can apply our AI systems, pour through all the genetic variants, and find out which ones are likely to cause a change to RNA biology and how that change could lead to the pathophysiology. So in that way, we can identify potential drug targets and novel targets. Briefly, how does it just take a step back now, overall for drug development, how does this AI workbench, which is what we call our system, how does it help us advance drugs across the entire pipeline? Everything from target identification all the way through to having a compound go into humans. The traditional approach to drug development is sequential. So you sequentially de-risk at each step. You have your drug developers who kind of make guesses at what's the best next thing to do. And then you'll test it experimentally. Maybe it'll take 6 months, maybe it'll take a year. And then you measure the experimental outcome, and it's basically trial and error. At Deep Genomics, what we do is we use these AI predictors plus our genomics data sets to make predictions up front for all of these different possible drug discovery outcomes. So before conducting a single experiment, we've identified whether or not there's a novel target. We've identified what the mechanism of action could be of a therapy. We've identified compounds that are predicted to achieve that mechanism of action. We've evaluated all of those compounds for their therapeutic window, so the balance between efficacy and safety. We've also used our AI systems to evaluate what the population size is. Can we expand the population by switching to a different mechanism? So all of those things can be done up front with our AI systems that are trained on genomics data sets. As you might guess, that makes it possible for us to do things that you just wouldn't be able to do in a traditional drug discovery company, and also do it faster and at lower cost. Another thing that I'm sure you're all familiar with and have seen in your own projects is the flywheel, the feedback loops, right? This is what research is all about. Research is about doing things and then learning and then getting better and then discovering new things that you don't know much about. What's cool here at Deep Genomics is we do that, of course, but we do that computationally as well. And so we generate huge amounts of data. The data is fed back into our AI systems so that the more we do, the better we get. So just to show you just one possible, we can start in the lower left, for example, we train predictors and create new AI systems. We have over 40 different machine learning predictors at Deep Genomics. We then use those predictors to evaluate targets and therapies, again, completely in the computer, make predictions for how that'll play out. We made over 2 billion predictions, every gene in the genome, 300,000 pathogenic variants. We have evaluated over 200 million novel steric blocking oligotherapies, 200 million, all computationally. All of that work is married with our experimental biology team. And so over on the upper right, we generate data. We've generated over 1 million efficacy and safety data points for over 250 genes and evaluated over 20,000 RNA therapies in vitro and in animal studies too. So all that data is fed back into our AI systems so that the more we do, the better we get. Out of that flywheel, of course, comes therapies. We have 10 preclinical today. We have 4 partnered. I will have 4 in the clinic by 2024. Something that's very relevant to all of us working on the genome is complexity, biological complexity or genomic complexity. And AI and our approach to using AI to solve problems of finding targets and molecules at Deep Genomics, it's not magical. It has limitations. It doesn't always work perfectly, as you might guess. And so our approach has been very methodical. We've started with simple biology and then built our systems, our AI workbench, to tackle problems for that simpler biology, and then moved on to more and more complex biology. So we started off with Mendelian recessive disorders, then moved on to dominant, and now we're looking at disorders of complex effect and also complex disorders with many variants that contribute to the disease. Whereas some approaches are to jump into the complex disorders right away and try to untangle the hairball, our approach has been to kind of start with simple, build the systems and the tools, those 40 machine learning predictors that enable us to have a good foothold when we approach complex disease. Just to give you a sense of the kinds of things we can do, those 40 machine learning predictors enable us to identify therapeutic targets, design therapies. We can predict safety and toxicity. We can identify new patient populations. We can predict other drug properties. What's really cool is once you have these tools in place, and again, this is sort of the power of genome and genomic analysis computationally, is once you have those tools in place, our researchers can do things with them that we hadn't even planned for them to be used for. And so, for example, one of our biologists, when creating a mouse model for one of our human genes, so needed to insert the human gene into the mouse, you can use different editing systems to do so. And the different editing systems will leave different scars behind in the DNA, like different mutations. And some of those could alter RNA biology in such a way that the system would no longer be useful as a model for human. And so that biologist used our splicing predictor, which I talked about before, to evaluate those different scars and figure out which editing system was most likely to recapitulate the human biology. We can use our SBOs to restore protein, to increase expression, and also decrease expression. So a very flexible toolkit as well. Where are we? So I've talked about the system, the platform, our approach, all of it based on genome biology and genomics data sets. The output, of course, is the drugs. And so this slide here gives you a snapshot of our pipeline right now. As I said, we're focusing on steric blocking oligos. It's sort of, you can think of it as a plug and play technology, which reduces a lot of risk, for example, in manufacturing and in safety. But we have 10 programs. We have 4 programs in CNS. We have 2 metabolic programs, and we have other partner programs as well. The lead cohort of programs, which will be in the clinic in 2024, include frontotemporal dementia, Niemann-Pick type C disease, Wilson disease, and refractory gout. We also have over 20 programs that are currently under assessment. We expect to have 28 programs in our preclinical research pipeline by 2024. All right, so I'm going to outline a couple questions that I think could be helpful for this community to think about in terms of translation. Before I do that, I want to briefly talk about our goals for 2024, as shown in this slide here. So a big part of what we're doing, this is roughly a third of our budget essentially, is to focus on building predictors for expanding into complex disease and also predicting SBO efficacy and also safety. So being able to predict ahead of time how our steric blocking oligos will play out when we conduct animal studies in mice and monkeys. We'll screen 100 genes for novel targets, 60 complex and 40 Mendelian, and we expect 80 of those to be successful. So the only way we could achieve that sort of productivity and that success rate would be using our computational analyses to look ahead of time at what's going to play out when we look at these targets. There's no way, of course, that we could take all 80 of those into the clinic ourselves in the short term, but we'll advance 28 of them into our preclinical research pipeline for validation and for compound identification. And then we'll partner a fair chunk of those out and advance 4 of them into the clinic ourselves. As time goes on, we expect to expand our clinical development capabilities and advance more into the clinic internally. But meanwhile, we'll conduct partnerships, and that's shown on the lower left. So a big part of the next few years is going to be to massively expand our partnership activity. That'll enable us to access the clinical development pipelines of other companies, but it will also bring in over $100 million of non-dilutive funding, which will help us run the company in the meanwhile. So that's what we're up to until 2024. I'll finish just by going through a few questions that I thought of about how the research community can have a high impact on Canadian industry or entrepreneurial activity. One thing that I think is exciting to see in Canada when it comes to the genomics community is the level of entrepreneurism. Most people in their research group come up with innovative tools, new solutions to problem, which to be honest is an entrepreneurial activity, right? It's not just research, it's actually producing tools that could help a lot of other people produce output. Now, whether you choose to take that tool into a company or keep it in the public domain or share it with researchers is one question. But I guess the question I'm focusing on here is just supporting that kind of activity. How do we translate research into tools, services, or products that can really help Canadian industry, Canadian society? So the first question I've outlined here, I think that most of us are familiar with this, is that some data sets are exponentially more valuable than others, okay? So not all big data sets are equal. And I think it's important to recognize this so that we can figure out which ones to prioritize. One approach would be to sort of say, there's 10 different categories in which we could create data, so let's just distribute the money across those 10 categories. I think that's unwise because I think certain categories of data are just much more valuable than others. And I would say the genome is a great example of one that's hugely valuable. The reason the genome is hugely valuable is I can compare genomes. I can compare two different human genomes. I can compare a human genome and a mouse genome, or a human genome and a yeast genome. The genome is something that's easy to compare. Comparisons are very useful for identifying genetic variants and how they impact biology and many other tasks. Whereas a data set that I think is harder to interpret and harder to make use of would be electronic health records. Electronic health records obviously contain a lot of valuable data, but the problem is, how do you compare two electronic health records, right? How do you measure similarity, and how do you leverage that to produce value? It's a tougher problem. I'm not saying we won't solve it one day. It's just not as clear cut than, say, sequencing genomes to find variants that are potentially causal for a disease. Another question I think is interesting to discuss is which existing entities can be used to produce valuable sequencing data. And for example, CanPath is just one example of an entity that is useful in terms of not just sequencing, but sequencing data sets that are useful clinically. And by the way, in all of these, I'm not taking one side or another. I'm just saying I think they're good for discussion. How should we direct public-private efforts? Okay. For example, we can imagine a scheme where Canadian biotechs request sequencing money, demonstrate their capacity to use it, right? And so it's up to the biotech to prove that that data is useful. And if it is going to be useful, then say Genome Canada could sign a check to generate a public data set, right? So make that data set publicly available. One thing I've learned at Deep Genomics, which is very much related to my previous life as an academic researcher, is when it comes to the genome, there's a lot more to be gained by sharing data than by sequestering it. There's so many unknowns right now. There's just a lot more to be gained by sharing it. Another question is, how can we obtain directed input from industry? So if we want translation, how do we set that up so that the biotech industry or other industries in Canada have a word when it comes to what kinds of projects would be most valuable? So, an example might be a Council of Canadian Biotech Entrepreneurs or something like that. So a committee to help identify big genomics data sets of strategic value to Canada. And I use the word entrepreneur there because it doesn't necessarily need to be a person in a company. It could just be someone who's like well known across Canada internationally for producing super useful tools. And then the last question is, what funding is available for translation? And Rob asked me to comment on this. And I have to say that there's just a lot of investors out there that want to pour money into this area, whether it's from the Bay Area or from New York City or Hong Kong, or Canadian investors. There's a lot of money for translation. And so if somebody has an idea, a system, a service, a product that they can build that's genomics based, my belief would be that if you can prove the value proposition, there will be VCs that are willing to pour lots of money into it. And so that would be my comment on that one there. All right, thank you very much. Happy to take some Q&A.